First, you have an exergonic reaction. What does it mean? Is straightforward. Any reaction that releases energy, that gives away energy, is called exergonic. In contrary, anything that needs energy, that requires energy, is called endergonic reaction. So you have an exergonic and endergonic reactions, and they are opposite. So if you know exergonic, you know what is endergonic. A coupled reaction, a coupled, it's straightforward again, is the association between an exergonic and endergonic. What does it all mean? Basically, the endergonic reaction, it requires energy. From where it gets energy? So when it's coupled, when it's added to the exergonic reaction, then it will happen. So the energy produced from the exergonic reaction will be used by the endergonic reaction to allow the overall reaction move forward. So if you look on to the right side, the big cogwheel describes the exergonic reaction, and they put it big to tell you this, it has a lot of energy. And the small cogwheel is the endergonic. So in order that the endergonic reaction happens, it needs energy, because as we said, endergonic reactions require energy. From where the endergonic reaction is going to get its energy? It's from the exergonic reaction. So if you look, the big wheel is exergonic and the small wheel is endergonic. So what is the summary of the slides? You have three types of reactions. Exergonic reaction requires, it releases energy. An endergonic reaction, it requires energy. You have to give energy in order that it happens. And the coupled reactions are reactions that have an exergonic reaction along with a endergonic reactions. These definitions are important to know, as I mentioned, because many questions have been asked before. Let's go to the next slide. So let's go into details. Now we are going to define, or to not to define, we have defined the exergonic and endergonic reactions. We are going to look at their characteristics. Again, these are very important. The characteristics are very important. Let's start with the exergonic reaction. We said that exergonic reactions produce or liberate energy. What about the free energy in the exergonic reactions? The free energy is the G that we said. G is for Gibbs. This is for your information. The person who found the this type of energy is called Gibbs. That's why they put G. So when the free energy of the final state is less than the free energy of the initial state, this is an exergonic. So what does it mean? If you look at this picture, on the y-axis, you have the free energy, G, as I mentioned, Gibbs, and the x-axis is the progress of the reaction. And as I mentioned before, and I expect you that you have in your prerequisite, you know what is a reaction, you know what is a reactant, and you know what is a product. So if you look at the top, A is a reactant. Let's say this is a glucose, and the product maybe is carbon dioxide. So you move in the exergonic reaction from A to B. What happens in this reaction? The initial state, A, is what it says. The final state is less than the initial state. This is the initial state, and this is the final state. And as you can see, the final state, which is B, we are talking about energy here, is less than the initial state, which is A. You see, this is top and this is bottom. The delta G, you see delta G. What does it mean, delta? Delta just, it means the change in free energy. G is the free energy. Delta G is the change of free energy. What does it mean, change in free energy? It means if you take the energy from A minus the energy from B, you will get a negative value. So if they tell you delta G is negative, automatically you know this is a exergonic reaction. As you can see here again, I will repeat the initial state, the final state, and the final state is less than the initial state, and the change in free energy is negative. So let's summarize what is an exergonic reaction. It releases energy. The free energy of the final state is less than the free energy of the initial state. Final state, initial state. The change in free energy is negative. The change is negative. This A minus B, it will be negative. We said it's a loss of energy, exergonic release of energy. So it will lose energy, and the reaction goes spontaneously from A to B. The word spontaneously is very, very important because many questions has been asked about this. Spontaneously, it means without any help, spontaneous. So the reaction goes from A to B spontaneously. It means we don't need to add energy. So now you have 4 points. You need to know these 4 points as far as the exergonic reactions are concerned. Okay? If we go to the negative, the endergonic reaction is completely the opposite. So if you know the exergonic reaction, which has 4 points, 4 bullet points, 4 characteristics, you will get automatically the endergonic. So the exergonic and endergonic are opposite to each other. So if you look at the endergonic, the free energy of the initial state is less than the free energy of the final state. You see, this is your initial state, is less than the final state. If you go back to exergonic, the initial state, the final state is less than the initial state. You see the curve, how it goes? Up and down. Here, up and down, but the initial state is less than the A, the final. Look at the delta G, change in free energy is positive. So if we take A minus B, you will get a positive value. So what are the 4 characteristics of A endergonic reactions? Require energy, the initial state is less than the final state. We are talking about energy, of course. The delta G, the change in free energy, is positive. So it needs energy. The reaction does not go spontaneously. And we need to add energy to the system. System, it means the reaction, to make the reaction go from A to B. So if you want to go from A to B, the opposite, we need to add energy. So you have 4 points to memorize. There is a third state called equilibrium. When the change in free energy equals to 0, the reactions are in equilibrium. It means there is equilibration here. If you have A is being converted to B as fast as B being converted to A. So if you have A, it goes very fast, converted to B. Then from B to A, it go very quickly. So when A is converted to B as fast as B is converted to A. So if you have a product here for anything, let's say glucose, it get converted rapidly to CO2, and then CO2 will be converted straight into A, the glucose. So this is basically what it means in equilibrium. So when the change of energy is zero, the reaction is in equilibrium, and A is converted to B as fast as B is converted to A. So these are the three states that what you need to know, their characteristics, and you need to know this. Please, in order to avoid to tell me, doctor, what we need to know in our exam, I am telling you now. So the student that they are listening should take notes and mention and write this. So Khadija, what do you think this, what type of this reaction? Khadija?
Yes, endergonic.
What type of this reaction is, if you look at it? Is it
Endergonic.
exergonic, endergonic, or equilibrium?
Endergonic.
Let's take
No,
you to
no.
this here. What about, look at this. You have energy, high energy here, initial state, and it becomes low energy. So when you have a high energy and goes to the low energy, what does it mean? High energy, initial state, goes to
Exergonic.
ahsanti. So it's exergonic reaction. Initial free energy of the final state is less than the free energy of initial state. So the free energy of the final, the product, is less than the reactant. Okay, now I will add to this, try to understand in the what happens in the real biological system, which is the cell. So the exergonic reaction the exergonic reactions are termed catabolism. Why is catabolism? Because it's a breakdown. Breakdown, it means oxidation. So you have a fuel and it gets oxidated. So this is catabolism, and I know this is again prerequisite. Students should know what is a catabolic reaction and anabolic. So catabolism, it means breakdown. Anabolism means building up. So the exergonic reaction, so basically this is the fifth point to add to the criteria of exergonic and endergonic reaction. 5 points. If they ask you whether exergonic reactions is catabolic or anabolic, you should know exergonic reaction is catabolic and endergonic reactions are anabolic. Let's look at an example. You see, you have here nutrient, let's say carbohydrate. If you eat glucose or fat or protein, you are degrading this. The carbohydrate will give you, will be at the end degraded into carbon dioxide, fats into water, and amine proteins. So basically you are degrading this, so it's a catabolism. If I'll give you a picture like this and tell you what is this, catabolic or anabolic. Just what you do, you look whether you say, oh, carbohydrate has been converted into smaller pieces. Big pieces, carbohydrate macromolecules are degraded into small molecules. So this is catabolism. This is a breaking down. The opposite, if I give you, let's say, amino acid, which are the building blocks of proteins, if I give you the sugar, which is the building block of carbohydrates, fat, which is the building block of proteins, nitrogenous bases as well of amino acids, and we are going to make macromolecules. What we mean macromolecules: protein, polysaccharide, lipids, nucleic acids. So we are building. So anabolism. The bottom line, exergonic reactions are catabolic. It means breakdown from big to small. And ergonic reactions are anabolic. It means from small to big. You don't need to know all this. The whole point is about knowing what is exergonic and the ergonic reaction as far as the energy is concerned. This is another, when we talked about the breakdown here, you see how, for instance, you have glucose, say 6H12 or 6, when it's oxidized, so oxidation, it means oxygen, what will happen? So this is the free energy. Again, we said the free energy. Can you see here how this energy is not produced all at once, but instead is building up? This is a building up of the energy. It's breaking down. You have a glucose. It has in its molecule, the molecule of glucose has a lot of energy. When we want to take out this energy, we add oxygen for oxidation, and then we start by step to take out, to makes energy. And as you can see here, there are many steps in order to get the final stage. So, and then the total energy is released. Now, why do you think we need to take energy by steps here? The answer is, if we want to make energy, oxidize the glucose at once, we will have a combustion explosion. So, and subhanallah, Allah subhanahu wa ta'ala has made this doesn't happen, and wisely the energy is building up by steps in order to avoid the combustion. So again, what is this slide about? It's an example how a breakdown of a molecule, and in this case is a glucose oxidation, how it's the breakdown of a oxygen, why there is of the glucose, why it's a breakdown, what we want from it. We want the energy from it, and this energy is synthesized by steps and not at once in order to avoid combustion. So now in these slides, we are talking about adenosine triphosphate as an energy carrier, and we call it high energy phosphates. Adenosine, adenosine, T for tri, it means 3. Phosphate is phosphate. So it's an adenosine, and when you add to it 3-phosphate. Energy carrier is carry something. So what carries the energy? So the energy in the cells needs to be carried. And who carries this energy is carried in the ATP, where in the bonds in the phosphates, and we will see. That's why it's called high energy phosphate, because the energy is stored in the phosphates bonds. So the ATP, it has a adenine, ribose, sugar, and 3 phosphates. So how can we make adenosine triphosphate? Is if you have a synthesis or breakdown. So you can make ATP by adding adenosine diphosphate. So if you have this molecule, which is an adenosine, add to it. So it has two phosphate, diphosphate. If you add to it an inorganic phosphate, it gives you an adenosine triphosphate. And if you have a breakdown of ATP, if you have an ATP and there is an enzyme called ATPase, and you know anything ends by ase, it means enzyme. If we want to cut it, it gives you the ATP, ADP plus inorganic phosphate plus energy. So this is just to tell you the molecule that carries the energy is in the ATP. So the ATP is the carrier of the energy. And how can we make the ATP? One of the way is to add an inorganic phosphate to an adenosine diphosphate to make adenosine triphosphate. So then this ATP, when we are cutting it to take out the energy we want, the cell wants to make the function, you end up by ADP, PI, and the energy. This is what we are after. This is the details of the ATP. As you can see, adenosine diphosphate plus inorganic phosphate gives you ATP. So you have this is what we call adenine. This is the base adenine, all this. You add to it a sugar, which is a ribose, and you add to it 3 phosphates, 1, 2, 3. It gives you adenosine, and we will see when we discuss the molecular side of this, how we name these compounds. But we should know at the moment, if adenine, you add to it a sugar, it becomes adenosine. So we call it adenosine because we have a base adenine plus sugar. So adenosine tri 1, 2, 3 triphosphate. And the energy usually is stored in 2 and 3. The energy. Again, this is slides. It reflects about, is talking about the way ATP, sorry, the exergonic and endergonic processes are synthesized. So if you look, the exergonic and endergonic reaction we talked about, what is their purpose? Is to make high energy compound, which is the ATP. So an alternative methods of coupling an exergonic to an endergonic process is to synthesize a compound of high energy potential in the exergonic reaction and to incorporate this new compound into the endergonic reaction, thus affecting the transference of free energy from the exergonic to endergonic. Okay, so we said that the coupling reaction, we said when we couple an exergonic reaction, let's say A is exergonic and B is endergonic, okay? Okay, that's, sorry. Anyway, so if we want to add endergonic, please exergonic, coupled reaction. Why we are coupling? Because the endergonic needs energy, so we need to put it in the exergonic with the exergonic reaction. what it says here, there is another way to another alternative method of coupling an exergonic and endergonic process to synthesize something that has an energy. We can do it differently by using the what we just mentioned, the ATP. So what we do is to make something that has a lot of energy, okay, to synthesize a compound of high energy potential in the exergonic reaction. So this is your exergonic reaction. If you make from it a high energy compound, let's say you make ATP from this, from A, which is exergonic, you get from it some ATP, then this ATP will be used for the endergonic reaction in order that the B works. So in this, at the first example, we use directly exergonic, endergonic, but we can make B work as well by generating what we call a high energy potential. In our case is ATP to move the B reaction. So in this diagram, as an example, glucose, this is a low energy and this is high energy. You start with the glucose, okay? So when you metabolize it, it means catabolism or anabolism reaction. At the end, the glucose, when you metabolize it, and you metabolize it by exergonic. What we said, exergonic are spontaneous and they don't need energy. You see, glucose as a product, sorry, as a reactant. You remember A, this is an example of A, and this is an example of B, the product. So A will be converted into B. So glucose, when it's oxidized, at the end, we end up by having carbon dioxide, water, and heat. What about the energy that is in the glucose? So is going to form our ATP. And this ATP will be used by B, for instance, by other reactions in order to be catabolized as well. So this is just to tell you the benefit of using the ATP from your reactant and to use this to move forward another reaction. That's why it's written here, protein. You can use amino acids from protein. So if you want to make amino acids, sorry, if you want to make proteins from amino acids, from where you get the reaction? Because you cannot make amino acids to protein, you need energy. That's why this reaction is an endergonic reaction. The endergonic reactions needs energy. From where we get the energy is from the ATP. From where we get the ATP? We get it from glucose. So, and glucose is an exergonic reaction, the oxidation. And this is what we are explaining. The bottom line, you have summarizing, you have two ways how to make reactions, endergonic reactions move forward. The first one, use straight and exergonic reactions. So the energy from the exergonic reaction will move the endergonic, or you use the ATP produced from the exergonic reaction, in this case is the glucose, and use it to move an endergonic reaction, which is in this case is an amino acids into proteins. This is the same picture basically. Model of ATP as universal energy donor. So if you see here the See, this is for instance, your glucose as an example before, but it can be lipids or anything. The oxidation will lead to carbon dioxide and water, and the energy produced here will be moving other endergonic reactions, for instance, this one and this one. So these will be moved. So when you eat food, high energy food like glucose, you produce, it's an exergonic reactions, and the energy produced as ATP will be moved the endergonic reactions like we've just seen amino acids to proteins. So this will move, this is a coupled reaction, and this is a different type of reaction. So basically this slides is exactly what has been mentioned previously. Again, this is a repetition of the if you remember at the beginning, we told you about what is the importance of the energy, free energy, and one of the and in our case is the ATP, and we said the ATP is the energy, high energy carrier. Why we need energy? Of course, we need it to make synthesis many molecules like proteins from amino acids, muscular contraction. If you want to move, you need energy, nervous excitation, mechanical work, active transport, heat production, all these need energy. From where? From ATP, and this is the origin is from exergonic reaction. So basically these slides and the other slides are the same. They are trying to tell you use the energy from exergonic reaction as an ATP to make all this work. So use of energy from an exergonic reaction to make an endergonic reaction. Okay, let's do this. So a biological system requires an energy so a bioenergetic system, we talked about what is a bioenergetic system. We gave example as a cell needs energy in order in the form of oxidable foodstuff. So basically any biological system like a cell needs food, needs, why we need food? We need to oxidize it. Oxidize it means adding oxygen. Why? We want to cut it down. We want energy. So we want energy from food. How are we going to get this energy? We have to degrade it. And degradation in scientific proper word in biochemistry is catabolism, to liberate energy. So how are we going to cut this food? Of course, we use enzymes. And I'm sure you've used many enzymes in the previous lecture to show you how enzymes work. Then this energy needs to be stored, synthesized, formed, like ATP, we need to get ATP, and we need to store it as a free energy. So the energy that we get from the food that we eat will be stored in the ATP, in the, as I mentioned before, in the phosphate bonds. Why we do this? Because we want the body as a biological system, the body as a whole needs this energy to do many functions. So again, this slides, it gives you example of the previous ones. It talks about we need high energy food like glucose. We need to oxidize it by oxygen. Why? To produce energy. Energy is ATP. So this energy will be stored in the ATP. Again, another explanation to how are we getting this ATP? When we eat carbohydrates, proteins, lipids, degradation of food stuff, catabolism, as I mentioned. Have you noticed that there are many things are repeating it themselves. And I believe it's good. So basically, this is, if you want to know what is a catabolism again, you see you have carbohydrates, and this is the units. When it's degraded catabolism into monosaccharide, protein will be catabolized into amino acids and lipids into glycerol and fatty acids. Okay. Remember, all these, what is the purpose of this? Is we want energy, we want ATP. So this goes through many reactions, and these reaction, I am sure, I believe you did you do the carbohydrate metabolism yet?
Yes.
Okay, so
Yes, Tahir.
thank you. So basically, you are aware what we are talking about. If you look at the carbohydrate, say Krebs cycles and all this stuff is in here. You eat in order to degrade the food using Krebs cycle. And if you look at your Krebs cycle, and then you will do protein catabolism as well and lipid, you will see that they all end up at a molecule called acetyl-CoA. And this will, of course, as I mentioned, enter the citric acid cycle or Krebs, and then electron transport chain, which is the subject of our lecture today, to generate ATP. So remember, all these these macromolecules will end up in the same, will give you the same molecule that enters cycles, and the end point is you want to make free energy. So if you look at this stage one here, and remember, if you put in your mind the picture of carbohydrate, for instance, that you did. Stage one, so you have a macromolecule, let's say glucose, macromolecule glucose of carbohydrate, as I mentioned, or protein or lipid, whatever. You want to cut it down into small units, as we did and we explained before, monosaccharide or amino acids or glycerol. So this is your first stage. You have a big molecule, it has a lot of energy. You want to get it into small pieces using enzymes. Then stage two, you get all these molecules again, following certain steps to reach the main molecule that will enter many cycles, what we call acetyl-CoA. So you reach to the acetyl-CoA. This is what you call intermediate of citric cycle, and some free energy is obtained. So once you get the acetyl-CoA, you get into the citric cycle that you did, Krebs cycle. What's the purpose? In this stage, you get only small amount of energy. You are not going to get much. The whole energy, the big energy will be you are getting this from the electron transport chain, which we are going to talk today. Remember, all these will end up by the citric cycle to get small energy, but the story will continue to get to get as much as you can of free energy of ATP from the electron transport chain. So the third stage, of course, this is what, again, the subject of our topic is to get into to get the most of the energy. So this will enter, most of the energy is obtained from the acetyl-CoA. So the acetyl-CoA is oxidized by the citric cycle to produce reduced enzymes. If you go to your Krebs cycles, and you see what is the end you know, always they give you a reaction. It means at the end of the cycle, what do you get in Krebs cycle or citric acid? You get NADH plus H and FADH and carbon dioxide, and it hasn't mentioned here a small amount of ATP. So what is NAD and FAD? This is like buses. Bus, you know, the shuttle. You know, when you go to the airport and you want to move from terminal 1 to terminal 2, you need a shuttle. So when you cut, let's say, glucose, and you want to take the energy from it, where do you put this energy, the shuttle? You put into the NAD and FAD. So these are those that they are going to carry the energy from your molecule, and take it to the electron transport chain to generate ATP. So reduced enzyme, these enzymes are reduced when they take electrons. Why we are talking about electrons here? The energy is in, as I mentioned, in the bonds that links for instance, the glucose. In this case, the acetyl-CoA at the end, and the energy itself is in the electron. So that's why we say reduced. If they ask you what is NADH and FADH2, is it oxidized or so reduced? They are reduced like this. Then they are going to get oxidized through the respiration chain. Okay, so they are reduced. Once they get into the electron transport chain, they are going to get oxidized. What does it mean, oxidized? It means the oxygen will be produced and the energy are taken out of them. Here they are full of energy. This energy is get it from the acetyl-CoA. When it reaches the electron transport chain, these shuttles, if you want to call them, or coenzymes, the right term is the coenzymes, will be oxidized to form water and carbon dioxide at the end. Of course, the energy. Do all the energy is at the end of the electron transport chain, do all energy is used? No. Small of it, sorry, not all energy is conserved, but few of it will be used in a heat and released as a heat to maintain the body temperature. We need heat. We need to get a 37 degrees temperature. From where we get this? We get it from once the energy is synthesized. Not all ATP energy is stored as ATP, but small amount is released to keep us warm. So you have acetyl-CoA here. It gets into the Krebs cycles you did in the carbohydrate metabolism. You get reduced enzymes. These are those I mentioned. Reduced enzyme are the NADH plus H and the FADH2. They are reduced. What does it mean reduced? It means they have energy in them. They have electrons added to them. And you get carbon dioxide. These reduced enzyme will get into the electron transport chain. So these reduced enzyme, the NADH and FADH2, will go into the electron transport chain. We will see where is the electron transport chain. And they get oxidized. You see, you have an oxygen here. When they get oxidized, what happens? The adenosine diphosphate is added to the inorganic phosphate to get ATP. And this is what we call phosphorylation. So I want you to understand and to have a general broad idea, and this will help you to understand what's happening. Basically, you are eating food. Let's say you are eating carbohydrate like glucose, lipids, and proteins. Let's take just one example, carbohydrate, glucose. When you eat glucose, why we need to eat glucose? Because we need energy. This energy in the glucose molecule, where is stored? Is stored in the bond C2H12O6. So within the bonds of this molecule, the energy is stored. Where? In the electrons. So the energy is in the electrons. So you have eaten glucose, and the glucose have energy, has energy in its bonds. Then we want to take out this energy. Then we have to cut this molecule, digest it by enzyme in our mouth, in our gastric tract, stomach. Then this molecule of glucose get into Krebs cycle. It will be transformed into acetyl-CoA. This acetyl-CoA will get into Krebs cycles. in this Krebs cycle, we get a little bit of ATP, not much. But the most important, we store all this energy from the glucose into a reduced coenzyme called NAD and plus H and FADH2. What happens next? We need ATP. So these NAD, H plus and FADH2 needs to go to what we call to electron transport chain, which is in the mitochondria. So they go inside and they get oxidized. What does it mean oxidize? It means we take out this energy by adding oxygen. So how can we get this? Basically by using the energy in the NAD and FAD, use this energy to transform the ATP plus PI into ATP. So we will see later how it's done, but this is the overall picture you should put in your mind. So whenever you eat food, remember, I am eating food, I want energy. The energy, it comes from glucose. Glucose will be cut out and get into a molecule called acetyl-CoA. This acetyl-CoA is going to get into Krebs cycle for what? Take a little bit of energy as ATP, but most of the energy is stored in the coenzymes called NAD and FAD. These coenzymes get out and go to the mitochondria. Why mitochondria? Because the electron transport chain is there. And in the mitochondria, where the ATP is formed by oxidizing NAD and FADH2. This is the electron transport chain. I'm not going to go into details, but just to briefly again explain. The substrate basically is the food you eat. Let's say glucose you eat. Glucose, again, this is called substrate. It will be, as I mentioned, get into the Krebs cycle and you get your NAD. The FAD is not mentioned here because most of the energy is from NAD, but we will get into details later. NAD is nicotinamide adenine dinucleotide. FMN is another coenzyme found in the mitochondria. So as you can see here, in each step, you are forming ATP. So using the energy to make ATP. And at the end, this is oxidation. You get the all bunch of ATP. So basically in the electron transport chain, if they ask you where the electron transport chain is, the answer is in the mitochondria. Where exactly in the mitochondria is in the inner mitochondrial membrane. So this is the exact place where the inner mitochondria, where the electron transport chain is. So don't get confused about this. We will get into it later in simple way. This is just to tell you how when you eat food as a substrate, how the NAD were mentioned here, NAD and FAD. As I mentioned, FAD is not mentioned here, but rather another molecule, but don't get confused. And it goes through a chain. Why is it called electron? Because they are the electron that they are going to carry energy from one place to another. Transport, because they are transporting the energy. Electron are transporting. Chain, why they are chain? Because they are steps. 1, 2, 3, 4. That's why it's called chain. So this is the explanation again, what I've just been saying. So energy-rich molecule, when you eat energy-rich molecule like a glucose, metabolized by a series, metabolize, it means cutting this food. What is the mechanism? It's the oxidation. And what is the end product? Your end product is carbon dioxide and water. However, when you are cutting this glucose, for instance, you will get some intermediate. These intermediate, let's say acetyl-CoA. Okay, the acetyl-CoA has the energy, then this energy will be given to NAD and FAD, as I mentioned before. And these are reduced, energy reduced NAD and FAD. Remember, energy reduced These are what we call coenzyme. And why they are reduced? Because they have energy. Once they give their energy, once they give their electrons, it becomes oxidized. Before these coenzymes and give their electrons, they are reduced. Once they give their electrons, they becomes oxidized. So NAD and FAD can donate pair of electron to a specialized set of electron carrier, collectively called ETC. So the NAD and FAD is not mentioned here, will give their electron to what? Donate a pair of electrons. It means giving 2 electrons to a carrier. These are the carriers. We are going to talk about them. Okay, the electron will go this way. And each time when it moves, it gives energy. As electrons are passed down the ETC, they lose much of their free energy. So once the electron goes from one place to another, when it moves from here to here, it liberates a little bit of energy. Then from here to here, let's say, just for the sake of example, if the electron has 10 unit of energy and it goes in this step, it releases 2 here. Then 8 is left. Then it moves here. It gives 2. 6 is left. It gives 2. 6 equivalent energy is left. Okay, so this is what it means. This is what it means. They lose much of their free energy. Part of this energy can be captured and stored by the production of ATP and from ATP and MPI. This is what we call oxidation phosphorylation. Part of this energy can be captured and stored by the production of ATP from ATP. So this will be captured when the energy is moved. It will be captured to be transformed into ATP by adding ATP to PI, ADP PI, ATP PI. So this is what it means. Part of this energy can be captured and stored by the production of ATP from ATP. The remainder of the energy not trapped as ATP is used to drive ancillary reaction and generate heat. As I mentioned before, not at the end, not all energy started from here is captured as ATP, but part of it, small amount, will be used for body temperature and ancillary reaction. What does it mean, ancillary reaction? They are, for instance, the calcium transport in mitochondria that makes heat. So there are many ancillary reactions that happens in the core, in the body, and the energy that uses from the energy released during the ETC. So this is basically what is the electron transport chain. Let's go slowly. So this is the mitochondria. Let's first know how the mitochondria is the structure. Its structure is very important. So the first point, if someone asks you where the electron transport chain occurs, the answer automatically in the mitochondria. This question has been asked before, and we tell the student where the ETC is happens, and whether in the mitochondria, cytoplasm, etc. So you should know it's in the mitochondria. Where exactly in the mitochondria? This is the answer again, inner mitochondrial membrane. Inner here. This is inner mitochondrial membrane. This is the mitochondria. This is the matrix like cytoplasm of the mitochondria. And this is the intermembrane space. Intermembrane space. So you have intermembrane space. Then you have the inner mitochondrial membrane. Then you have the mitochondrial matrix. So this is the overall. Here it will make it clear to you. This is a mitochondria, inner membrane, outer membrane. So you have an outer membrane, you have an inner membrane, then you have an intermediate space, this one, and the matrix. So again, this is why I'm going forward and backward, because it's inner mitochondria. Don't get confused with outer membrane or intermembrane space. Is the inner membrane, outer membrane, then you give the intermembrane space, then the inner membrane. So we are focusing in here. Okay, so this is our inner membrane. So when you have food, when you eat food, it becomes acetylCoA in the Krebs cycle. NAD and FAD will carry electrons and then comes here to give the electrons. As you can see here, this is our NAD that carries a lot of energy in the electrons. And we have here complexes. See, you have complex 1. They haven't mentioned complex 2 because this is related to FAD. And FAD doesn't produce much, is not involved at the beginning. So that's why it's not usually in textbook you won't find it. So you have complex 1, complex 3, 4, and this is a 5. And then this is where your ATP is formed. So as you can see here, NAD is full of electrons, and here you are. You start the electron. So when the NAD gives its electron, what we said here is reduced. Is given the electrons, it becomes oxidized. So now the electron is captured by complex 1. Then the complex 1 will give it to complex 3, and we will see what carrier. So, and then complex 3 will give it to complex 4. And as you can see here, how the electron flow is going. Until it reaches complex 4 here, and then there is a oxidation and formation of water. So it starts here by NAD full of electron of energy and end up or formation of water. And then you will see later what happened in this stage. All the energy gathered here will be used to turn this wheel to form ATP by adding ADP to PI. So this is just an overall. Okay, apologize again. This is not my lecture, Yani. So forgive me. There are things that, although I checked the lecture before. So the release of energy, of free energy. This is the get energy. This is the free. You remember when we started the lecture, we introduced the free energy. This is why we are talking about always the free energy. So the release of free energy during electron transport. So this is what I mean. Move of energy. Free energy is released as electrons are transferred along the ETC from an electron donor, reducing agent or reductant, to an electron acceptor, oxidizing agent or oxidant. Again, these are, as I mentioned, the basic and prerequisite. You should know what is oxidant and reductant. Here, the free energy is released as electrons are transferred along the ETC from electron donor, which is NAD and FAD. So they are reducing agent or reductant to an electron acceptance. These are the carrier that we are going to talk about. The electrons can be transferred as, again, don't worry about these details. This is I told you when there are things that I believe it's not that important. Don't worry. Remember that the energy is transferred from the electrons. There are different ways how the electron is transferred, but I believe it goes into details. You don't need this information. So the electron can be transferred on such and such. So it is transferred, the electron transfers in different form. Again, this is just a point where to remind you what is redox pair. So basically this slide is what we call prerequisite. Again, you need to know what is oxidant, reductant, redox pair. Redox, it means reduction oxidation, because all electron transport chain is about oxidation reduction. So you need to know when you are learning, you need to know what is oxidation reduction. If you want to understand what is the electron transport chain, you need to fix this in your mind. What is oxidation reduction and what is oxidant reductant? So you have to go to your chemistry course before and make this very clear. So as far as we are concerned, we are talking about oxidation and phosphorylation. So oxidative phosphorylation occurs in the mitochondria, as I told you before, and even I told you exactly where the place. So the transfer of electrons down the ETC is energetically forward because NAD is a strong electron donor and the oxygen is an avid electron acceptor. Avid or avid, it depends on the way you pronounce it. It means it's really want the electrons. What does this mean? We are talking here. This is the NAD. NADs like to give electrons, and oxygen here in complex 4 here like to take oxygen. So that's why the electrons are moving from this side to this side, because this like to give and this like to receive. So the flow of electrons from NAD to oxygen does not directly result in ATP synthesis. Again, when this flow goes from here to here, does it make automatically oxygen ATP? The answer is no. It needs what we call phosphorylation, and this is the phosphorylation we are talking about. Why phosphorylation? Because we are adding phosphate. So even though the electrons are moving from this to this, doesn't mean we are going to get ATP unless this part here is involved. And this is what we call phosphorylation. This part is oxidation. Why? Because we have the end here is oxidation. And this part is phosphorylation because there is a phosphate adding to ATP. That's why we call it oxidation, phosphorylation. They need to work together to produce ATP. Electrons removed from NAD and FAD are passed along a series of carry cytochromes to produce ATP. Now we are starting to get into more details. So as I mentioned, to go from here to here, from here to here, you need cytochromes. These you need carrier or shuttle that I mentioned before. These are called cytochromes. There are other carrier we'll talk about later, just to mention them to you. So the first information, these are called cytochromes. So what is a cytochrome? Is a carrier. The carriers, I hope you know what is a carrier. A carrier that takes electrons from NAD and give them to, they carry electrons from one site to another, from complex 1 to complex 2. It's like a bus, as I mentioned. A bus will take electron from this complex to this complex. It's called cytochrome. From this to this is cytochrome. As I mentioned, there are other carrier involved, and we'll talk about them when they come. Now, we said at the beginning, we have two coenzymes. You remember when he said, when you eat glucose, acetyl-CoA, you get into Krebs cycle, and in the Krebs cycle, you get NAD and FAD in a reduced form. It means have energy. Now, remember, we haven't mentioned FAD. As I told you, we didn't mention FAD because it produces less ATP. This question has been asked before. So bear in mind, if they ask you how many NAD, how many ATPs NAD produce, you say 3. If they tell you how many FAD, how many ATP produced by FAD, 2. Why? And this is the point I explained before. The NAD, there is another complex here. Number 2, as I mentioned, is not written here. The NAD will enter 1, 2, 3, 4, 5. The FAD will go from 1 to 3. And it misses complex 2. That's why the energy is less in FAD than NAD. I repeat, NAD produces 3 ATP because it goes through complex 1, 2, 3, 4. FAD, it produces 2 ATP because it misses complex 2. Now, what is chemiosmotic hypothesis? Basically, this is, again, is a hypothesis, nadariya. Okay? It's just explain how this free energy that comes from the electron transport chain produces ATP. So, how the electron is moving from this explain the formation of ATP. So the way we are explaining the move of electrons through the electron transport chain to form ATP, this explanation is called hypothesis, chemosmotic hypothesis. So the NAD and FAD are proton H plus from NAD and FAD are accepted by O2 to form water. So this is the details. If someone asks you from where the water is formed, you tell him, of course, you get H plus produced here. We will see later. When the electron is moved from one side to a side, from one side to side, what will happen? The H plus will go from the matrix into the intermembrane space. And then at the end, you see, if you look here, you see, when the electron is moving from this place to this place, there is an H plus was here in the matrix, goes into the intermembrane space. So this question has been asked before. When the electron moves from complexes, either complex 1 to 3 or 1 to 2 or 3 to 4, the H plus goes from mitochondrial matrix or towards intermembrane space or the opposite. So remember, when the electrons are moving from one complex to another, the H plus proton hydrogen plus proton goes from the mitochondrial matrix to intermembrane space. Again, from here to here, H plus is getting out here and is pumping out. Can you see? So if you have, let's say you have seven H plus here, an example فقط, you will get them all here. So at the end, at complex 4, you have a lot of H plus here. You will have lots. What will happen? Then suddenly, this H plus is a lot of energy. What will happen? They will go back again to their mitochondrial place. So see the H plus? All this H plus gathered here will go back. So when through what we call complex 5, that has what we call FC domain in the intermembrane space and F1 domain, which is in the mitochondrial space. Is important to, when I read it later on, I'm not going to go back to it, so we save time. So again, this H plus, when it gets in through complex 5, a lot of H plus will enter again. What will do? It will turn this wheel. This is a wheel. So this will turn. When it turns, what it does, it takes the adenosine diphosphate, it adds to it inorganic phosphate to form ATP. Then the ATP will go outside the cell, go to other cells to do other reactions. Then how are we going to get the ATP here? The ATP that is found in the intermembrane, it comes from, of course, the cytoplasm and then enter the mitochondria and get into this in the mitochondrial space. So this picture is summarizing what's happening. You have electrons goes from one complex to another. The H plus will enter the intermembrane space from mitochondrial matrix. Then there is a buildup of H plus. At complex 4, the H plus get into the